Hello everybody and welcome to the first IBH Seminar of 2025 and Happy New Year to everyone also. I'm starting the new year with a new platform, and instead of a WebEx meeting, we are trying out a WebEx webinar. The layout will be very similar. I'll hand over to Gwen soon, who'll introduce Rudiger, and then he'll give his presentation. And if you have any questions, please put them in the chat, or if you would be keen to ask your own question and be on video and to unmute, just put your hand up and we'll make that possible at the end of the seminar. The last thing is, when the seminar ends, there'll be a feedback form, which you can please let us know what you think of the new platform, positive feedback only if possible, and then we'll take things from there. So I hope you enjoy the seminar, and now I'll hand over to Gwen. Thanks.
Thank you, and thanks, Rudiger, for coming today and waiting over the technical difficulties. And maybe you have to do your presentation now with like a green circle on your slides if you cannot get rid of it anymore. So it's my great pleasure to introduce Rudiger Simon today. He studied biology at the University of Cologne, where he did his PhD on transposable elements in maize. Then he worked as postdoc with Enrico Cohen at the John Innes Center on flower development and snapdragon. Spent a short time at the IPK in Gartesleben, where he worked on nematode resistancy in tomato. And then again at the John Innes Center with George Coupland, working on the control of flowering time in Arabidopsis. He started his first own lab in Cologne. He started his work on the stem cell regulation in plant meristems. And in the early 2000s, moved with his lab to Düsseldorf, where he's now a professor for plant developmental genetics and still focused on stem cell regulation in plant meristems, in the shoot and in roots. Started with Arabidopsis, and for the last 4 years, also working on barley. And I say a few years because it feels like it was yesterday, but it's probably not a few years, it's actually long ago. And that's why we invited him today to present some work on the combination of single-cell RNA-seq with spatial transcriptomics. And we're very excited about that. The stage is yours.
Thank you, Gwen. Thank you to all for the invitation, to all the participants for participating. And sorry about some technical issues. So I will have to do it like in the old days. Next slide, please. So next slide, please, which is sort of the intro slide. Oh, there we are. So you heard the title already. And as Gwen indicated, my lab was focusing mostly on studying development in Arabidopsis with a special focus on meristems and on the behavior of stem cells in the meristem. And that's why I have this kind of starting slide here. And some years ago, we were heavily influenced by colleagues here at the Heinrich Heine University in Düsseldorf, specifically by Maria von Korff, who has worked on barley for a long, long time already and was an expert in that. And she said there are so many developmental problems that still need to be addressed in barley, that the field could actually benefit from some extra input. So we got really curious about the barley inflorescence architecture because, and I hope you can see my pointer here. Oh, maybe, yeah, no, because we have so many different types of meristems here, which undergo a sequential differentiation steps, and all of this in a very, very coordinated fashion. So if you take such a young inflorescence, a spike here from barley, and you take it from several plants, they look very, very similar. It's a very, very reproducible process how all these meristems here undergo their proliferation, cellular proliferation, specification of different organs later on the floral meristems, and as they grow out, or some of them arrest and some of them undergo some cell death. So it's a very, very intricate network of interaction, cellular interaction, but also within a meristem, but also then interaction apparently and coordination between meristems. And we had studied in Arabidopsis behavior of stem cells, and on the center of the slide you see a confocal image, 3D actually, of a Arabidopsis inflorescence tip. And we had learned there that signaling pathways mediated by phytohormones, but mostly also by peptides and receptors and by transcription factors, coordinate these developmental progression. And for us, it seemed like the grass inflorescences, and for our example now, barley, really poses some extra challenge with this coordinated development of so many different meristems and meristem types. So if you can go to the next slide, please. And here we just have a comparison of some inflorescences from different cereals that are also important for agriculture. And another aspect which became apparent to us is that cereal architecture is also an important factor that controls overall yield, because the architecture in the inflorescence controls ultimately the number of kernels of seeds which can be made by a plant. And on the next slide, there are some scanning EM overviews. If you just press the button, please, you will see some scanning EM overviews of wheat, barley, and maize inflorescences. Can you? Yeah, there you go. What we notice here is that the architectures in these different inflorescences are in some ways they are similar. We have these rows of primordia which are being initiated, starting from the inflorescence meristem at the tip and then going downwards. So we have nice developmental progression going from tip to the base of the inflorescence here. And we see, of course, differences then which are species specific. If you go to the next slide, please. The approach that we took, and I wish I would know how to get rid of this green button now. The approach that we took is the following. We thought we wanted to achieve a mechanistic understanding of meristem development in grasses, with focusing on barley here in the first case. And the approach that we took is basically starting from our knowledge from Arabidopsis, that we could use genetics, either use already available mutants, edit candidate genes based on prior knowledge, using transgenic approaches, this is indicated here in this green box, modify and first learn how to analyze, and then characterize the phenotype of meristems at different states, then study our mutants, phenotypes them in detail, development, morphology, anatomy, signaling pathways, and an outlook for the future. We will also want to do that under different environmental conditions, with light and temperature being sort of the key factors that we will vary. We then went into I call that multi-dimensional transcriptomics. That means we combined a lot of different transcriptomic techniques which are nowadays available, ranging from single-cell RNA-seq to spatial transcriptomics, tissue-level RNA-seq, so where we just cut off whole tissue bits, and then explore developmental trajectories from that. And RNA data are important, but of course we want to understand ultimately gene regulatory networks, with the idea in mind that we can then better understand and also manipulate inflorescence architectures. And for that, we need to know the cis regulatory elements. And there are a couple of techniques out there in the field which can be used, TechSec, StarSec, and we are focusing on a technique which is called MOA-seq, based on micrococcal nucleases. And now what is still sort of shaded in gray is the analysis of molecular networks and exploring dynamic functional models. So also puts this whole process of inflorescence development into a computer model. And that is hopefully something we can start to do this year, and then come up with some first models in the very near future. If you go to the next slide, I can show you how we went on from that. So on the left, you have a color-coded scanning electron micrograph of a barley inflorescence. And we labeled here in blue the inflorescence meristem tip itself. And I think you're all familiar with the overall development of inflorescences. I just want to run you very, very quickly through the different meristem types that we are addressing. So first, a central spikelet is being formed that splits up, or triple spikelet meristem that splits up into central spikelets and two lateral spikelets in these two-rowed variety of barley that we are using. Then we have a floral meristem, which is subtended by lemma primordium. Further down, we see gloom primordia. There's a tiny structure here called the rachilla, if you pronounce it in German, which is not developed extensively, but this will be addressed in a later seminar in a few weeks' time by one of our co-workers here, Isaia Vadaniga. And then we have the development of flower meristems with floral organs and so on. So we see already just by the different colors that have been used here, there are a lot of different organs and structures and meristem types which are developed in a progressive manner, can be identified already at an inflorescence here at the so-called Waddington stage 3.5. So to get a very unbiased view of the processes that are ongoing here, we started to do single-cell RNA-seq experiments. So we harvested those tips, protoplasted them, made sure that the protoplasts that we identify or isolate are really representative for the whole structure. So for that, we used a transgenic barley line, which expressed gene, which expressed the fluorescent protein in the inflorescence meristem and a few other meristems, and we made sure that we find these protoplasts for all different meristem types, which different size, in the samples that we isolated. We tried a couple of different methods, among them 10x genomics and BD Rhapsody, and for us, BD Rhapsody, where these single protoplasts sort of are allowed to sort of settle by gravity into microvels and be thereby separated from each other, was the best method to use, where we obtain the highest number of viable cells. The next steps then are single-cell RNA sequencing. You of course have to filter out for protoplast-induced genes, so that requires a number of controls that you do, and then you can start analyzing the single-cell data that you obtain by clustering. If you go to the next slide, please. The clustering itself is a method where there are lots of different computer softwares for there, which basically is asking which cells are, after you determine the individual expression profiles, are more similar to each other, and then they're aligned in beautiful UMAP plots, which you will also see later on. Whether you can separate cells with different identity from each other or not depends very much on the settings, how you allow your computer programs to run. And one example is shown here. So there's a parameter called cluster resolution. And depending on how we set this parameter, cluster resolution, we can either separate only meristems from each other, or we can even separate different cell layers within the meristem. Example here on the top, L1 from the floral meristem epidermis can be separated from the rest of the floral meristem, or we have here the L1 separated out from the floral organ epidermis at high cluster resolution, while at lower cluster resolution we're not able to do that. The danger there is, of course, that you're creating some artifacts. You can create clusters which are not really meaningful if you look at the development of your tissue. If you go to the next slide, please. Good. Here's a typical UMAP plot on the left-hand side, and you have to keep in mind that what we have here with these UMAP plots is basically a multi-dimensional clustering or analysis of cells and their proximity to each other, mapped down on 2 dimensions to make this all visible. And whether you get now 25 clusters or 30 clusters or only 10 clusters depends to a large extent on the settings, how you really adjust your analysis programs. So these clustering is all very nice, and then with the help of some genes that you know already, famous marker genes like knotted, for example, which is expressed in meristematic tissues, you are able to sort of assign specific identities to the cells that you have separated now out into these different clusters. What we try to do is to say we want to, we had here something like 25 clusters or so. We thought, okay, let's take 3 or 4 genes which are sort of very, very specifically expressed in only one of these clusters. So let's take these genes and let's test by RNA in situ hybridization where these cells are actually coming from in the original tissue. And selection of this gene is not sort of random, but rather we selected some, of course, which have a high specificity for the individual clusters, which show a very, hopefully a very dynamic expression range between different cell types, and that show an at least reasonable expression level. So if your distinguishing feature between one cluster and the other one is a gene which is only expressed with one RNA or no RNA molecule at all, then this is not too meaningful and not really productive. So if you go on the next slide, please. Here's an example of what we did. To the left, you see again the inflorescence, barley inflorescence with color-coded with the different meristem types and organ types. And here are a few examples of some of the genes, cluster-specific genes that we took for analysis. So I don't want to go into any detail here, just enjoy the different colors here. But I think what you can see already is that the different genes are very often expressed in a very highly specific pattern. If you go, for example, to the very right, the gene which is whose signal or detection in the tissue is actually indicated here with yellow dots. That's a gene, Matchbox transcription factor, and you see it's only expressed in sort of the maturing or the later stages of floral meristem development. And the techniques that we used here is actually a single molecule RNA FISH technique. In this case, that is offered by several different companies and different systems. This one was called molecular cartography by the company Resolve, but there are also other companies which we do that in a similar manner. So what we did here is we generated a total of 100 probes for 100 different genes and hybridized them on tissue sections. And then you can use different colors to sort of later on display the detected RNAs with subcellular resolution for the different genes that you're testing. We did a lot of controls on this, which showed pretty nicely that the number of dots here, which we detected in a given cell, is actually representative to the overall gene expression levels, which we could prove by other method. So in principle, what we can do now is count the detected dots in each of the cells on the tissue section, and thereby get a nice overview on a total of about 100 genes, which are characteristically expressed in each cell, or which were characteristically detected then in the individual cells. If you go on to the next slide, please. When we go back to our single cell analysis, we can here define developmental trajectories using pseudotime to identify different gene modules. So if you here, for example, we were focusing on Matchbox genes, which are transcription factors, very often key regulators in developmental processes, and there are about 120 Matchbox genes, proteins encoded in the barley genome, and 90 of these Matchbox genes were detected to be expressed in our single cell expression data analysis, and 31 of them were abundantly expressed, so expressed at really high levels. So during developmental progression here from the top left, sort of uncharacterized meristem, to the bottom right, where we have specific floral organs, for example, stamens, we can basically describe here a pathway of cellular differentiation, or so-called developmental trajectory. And that is also indicated here on the right, where we display on the inflorescence the expression pattern of a number of different Matchbox transcription factors, and which is here nicely represented in this totally independent experiment by the single molecular RNA FISH data. So if you go to the next slide, please. Now the question that we were asking is, we have in principle here two different data sets of two different qualities. On the left is just the typical UMAP plot. There we had protoplasted plant tissues. We obtained a certain number of cells, and for each of these cells we achieved information on average on about 6,000 expressed genes. In total, if you add up all these different protoplasts, we found expression here, or expression information, for almost 48,000 different gene models. And that includes the 81 so-called anchor genes. We aimed at 100 initially. For 81, we got reliable data. 81 anchor genes, which we also had, where we had prepared probes for the single molecule RNA fish experiment. On the right-hand side, we have a typical tissue section. On each of these tissue sections, we had whatever, around 20,000 cells or so. And here we have the precise location, or the localization for each of these cells. And we have expression levels and patterns for 81 anchor genes, but not for the 48-something thousand other genes. So how could we bring these two types of information together? So the first thing that we did was, if you go on the next slide, please. So what we did, we combined the clustering. So we took all the cells that were identified in our tissues, in the sections, in the single molecular RNA fish, combined that with the single cells, with the cells isolated in the single cell RNA-seq experiment, did kind of a combined clustering, and then basically identified cellular similarities using the 81 anchor genes as a key. So in other words, we had on the right-hand side localized, we have specific locations for a cell which expresses where we have information on the expression level of 81 different genes. With that information, we went into our combined clustering and asked, what are the most similar cells? And these most similar cells are then most likely localized at exactly the same position or at a related or nearby position to the cells that we identified on the tissue sections. But what this repositioning of a cell from a single cell experiment brought with it is information on all these thousands of other genes which were identified as being expressed in the single cell experiment. Thereby, we could basically map our cells from the single cell experiments onto our tissue section and get a combined set of information out of that. What this is called is, in principle, there's some mathematics behind it and a lot of statistics behind it. So this is called gene expression imputation. I want to show you a little bit what the quality of that data set is. If you go to the next slide, please. Okay, first of all, we have generated or integrated all this data into a new database, which will be open to the public very soon as we have the paper submitted to bioRxiv. We are still in the final process of writing. Then this will be open to the public and for everybody's use. So what the database basically integrates is, if you look on the left side, there are these typical clusters. So all the information on all the cells which we identified in single cell experiments are integrated there. Then we have the information of the tissue sections, and this information is now linked. Sadly, I cannot show you that now live on the database because my laptop computer just doesn't do that. The data sets are a bit too large. But what I will show you with a few examples on the next slide is the following. You can basically now click on any cell with your mouse in this data set on these tissue sections, and then on a display below, you will get information on the expression level imputed for all barley genes which were available in our annotated databases. You can also filter. You can ask, for example, where is Matchbox gene so-and-so expressed? You can type that into a window here, and you will get a presentation either from real, let's say, real single molecular RNA fish data, or from the imputed data, and I will show you the quality of that on some of the next slides. And on the right-hand side, there is an example where we had asked for the expression patterns of two different genes. One of them is the knotted transcription factor, depicted here in red and orange colors, and that is nicely expressed here in the meristematic tissue and also in the vasculature, but not where new organs are about to be initiated. And at the same time, there's another transcription factor here depicted in blue, of course, also with kind of, how to say, color code with different colored intensities, which I think was VRS4, but don't name me for that. I'll show you a few other examples later on. So that's the kind of output that you can get, and you can do a few other things more with this database. If you go to the next slide, please. Okay, quality and limits of gene expression in imputation. We used a method, or we used cosine similarity to identify or to show the similarity between different cells. Then we average gene expression values. They were weighted based on the similarity detected for the 81 anchor genes. We filtered out protoplasting effects, and we in the end got expression values for 48,000 gene models and for more than 20,000 cells per section. And the one quality parameter is basically the cosine similarity average for the imputed and measured cell gene expression levels, and that was higher or about 0.84, which is a very, very good value, I think, compared to data sets which I've seen from other people's experiments, not in plants, but in animals. But we are still working on methods how to improve this further. I give you a few examples if you click on the next slide. It probably tells more than 1,000 words. So on the left-hand side, you see the tissue sections that you're all familiar with already. So that's the inflorescence here. And here we have a real fish experiment where we used the probes for a specific gene. I can't remember which one. And you see here the expression pattern of it. Now, what we also did as a kind of control, we took this gene out from the data set, but imputed its expression using all the other data that we had from the, let's say, 80 other anchor sequences and from the single cell data sequences. And what you have here is the imputed gene expression pattern. So compare this pattern here of expression with the one we really detected in the fish experiment, and you notice there's a very, very high similarity between these two patterns. This is here for inflorescences at one stage 3.5. And on the right-hand side here, we have it for the vegetative shoot apical meristem. Again, a fish experiment with one probe, and then the imputed pattern. So where we took out the real information from that gene and just imputed, predicted the expression pattern. And you notice there is a very, very high similarity. We're also missing some cells, which in the real data set do express the gene, but these cells here, the expression in these cells were not possible to detect by our calculations. But I think at least it gives you a very, very good impression of the quality of this imputation method. Now, it's not perfect. There, for some genes, it didn't work at all. And there are two examples shown on the next slide. Next slide, please. So here, that's why it's sort of with a green background. These are the nasty ones. So here on the left-hand side, we have again the fish experiment and the imputed expression pattern, and that didn't work at all. And also that is for the inflorescence meristem. On the right-hand side for the vegetative shoot apical meristem. The fish experiment and the imputed pattern look very, very different. These are some of these rare cases. And we noticed that both of these genes are actually very, very sensitive to protoplasting artifacts. That means that during the protoplasting step that we did a couple of controls there, these genes are changing drastically in their expression patterns. So those ones are really difficult to detect, but we already have some ideas how we also get better information on these type of genes. So what can we do with all this? If you go on the next slide, please. So here's again another comparison. On the left, we have the molecular cartography data, the real data from a single molecular RNA fish. And then on the right side, you see a couple of examples where we have now the expression patterns which come from the imputation experiment, so from the BARVISTA database. And you notice there's a very, very high similarity, and there's a very, very high predictive value of our gene expression imputation experiments. If you go on the next slide, please. What you can do is something which I would call virtual microdissection. For example, you have the question, or the question might come to your mind, what actually happens during progressive development of those meristem types, young meristem types, at the inflorescence of barley. And on the left, I've just indicated, I've used the BARVISTA tool to just mark in red some of these cells that I find interesting. And let's say a planner experiment where I want to compare gene expression patterns between the first primordium and the next primordium and the next primordium and so on, and ask how do gene expression patterns in these cells progressively change during development. Or maybe I have a very specific question in mind, and I would like to ask, what is the difference between the epidermal cells at a position where a primordium is formed compared to a position where no primordium is formed? So what I can do with the BARVISTA tool is label those cells and then download all the expression information for 48,000 genes for those cells that I've labeled here. And then undergo kind of subclustering, so next step of clustering to look for similarities and differences in gene expression. What you get as an output is something which I try to indicate here on the right. You basically get a table. Each of the cell on the tissue sections has an identifier, so it has an individual number. And under this number, gene expression values for all genes that are annotated in the barley genome are stored. So that table, which I indicated here, is actually incredibly long with like 48,000 columns. So it didn't quite fit on the screen. Can you go on the next slide, please? Here's one example where we did exactly that. So we asked what is the difference between these different primordia types here from P1 down here in this experiment to P19 on one side of the inflorescence. So the plot here indicates the expression changes in specific Matchbox transcription factors, which we took here as an example. We can, for each Matchbox, for each gene or each transcription factor in this case here, we can, here in a column, we can ask for its expression level in how many cells and at which level during this progression here from P1 to P19. And what we noticed is that if we look at the overall differentially expressed genes by comparing P1 to P3, P3 to P5, and so on, we notice there's kind of an inverted hourglass model where we have the highest sort of difference in expression patterns here in restricted here to transcription factors at the level here at around the stage P5. Here there's highest diversity compared to the neighboring primordia, and that goes down here. And actually, if we look at the developmental stages at the inflorescence, these meristem types here are very much more alike phenotypically, and there are more radical changes here going through these early steps from P3 to P7, which is nicely reflected in the expression patterns of these transcription factors. And of course, these were the ones, next slide, please, which we had already studied in our single molecular RNA fish experiment. So you can now, here's the predicted data, or the ones which extracted from BARVISTA, and these are the real, let's say, the hard data from single molecular RNA fish. And if you go through the different transcription factors highlighted here, you will find exactly the correct pattern. If you go to the next slide, please. We can also use that to identify organ-specific expression, for example, again, of Matchbox genes. There's the famous A, B, C, D, E model by now for organ type specification in the flower. And here's just an indication of that for the different organ types in the barley flower and the combination of different Matchbox transcription factors, which are specifically expressed in these different organs. Next slide, please. We have also extended BARVISTA to mutants. So we have basically done the same procedure that we did for wild type barley, also with a couple of different mutants, and that is an ongoing process. We're adding more and more mutants to the database. We can use the expression information we then get for tissue type fingerprinting. On the left-hand side, we have here Golden Promise Fast, a wild type, and indicating the different meristem types, and then a couple of different genes that are expressed here in these meristems. And on the next slide, you will see a double mutant compositome 1, compositome 2. Press the button, please. Thank you. So that's a COM1, COM2 double mutant, which shows the different branching patterns. So instead of just determinate floral meristems. Here we have an indeterminate branch developing, and that is also reflected then in differences in the expression patterns. Here we have specific genes, like here's the yellow one, which is popping up again and again and marking characteristically those branching new meristems. So we can use it to characterize mutants. If you go for the next slide, it's the same example in principle, but here we have used created kind of super clusters for specific organ types of specific tissue gene expression patterns. So what you have here is the different colors do not now indicate the expression of a single gene, but rather a complex expression pattern which is specific for a certain cell type. For example, here bright orange, that is sort of meristematic, and red, that is of indicates the ground tissue, and so on. And we can then use that in a clustering. And if we now compare the wild type scenario, golden promise, wild type, just look at this cell group here. We did a similar single cell analysis for the double mutant. You notice that this cell group here is much smaller. So it appears that this cluster here is underrepresented in the mutant. And indeed, that cluster is derived from cells which undergo floral organ specification. If you go on the next slide, please, or press the button again. We can also do this comparison now by basically labeling on BARVISTA just the tip of the meristem golden promise, and ask, you know, what's the differences in gene expression, high level gene expression between golden promise tip and here a later stage developing floral meristem. And we see here in this scenario where we have used certain filters, we find more than 600 differentially expressed genes at high levels. And if we do that in the mutant, where instead of a floral meristem here, a branch apparently is being produced, we find no high-level changes in gene expression patterns, indicating that that structure can be characterized at an early stage already and depicts shows exactly the same gene expression pattern as the inflorescence of the wild type. Go to the next slide, please. I think I have to come to an end very soon. Just another example, virtual microdissection and subclustering. Here we were asking whether we can, whether cells which are likely to form the next primordium are already transcriptionally distinguishable from inflorescence meristem or from older primordia. So here we compared inflorescence meristem to the P0 state and to the P1 state, which is plotted here. And we can clearly identify groups of genes which are specifically expressed or upregulated during these different developmental progressions. How many more minutes do I have, Malcolm or Gwen?
I think 5 or 10.
Okay.
Because we have started a bit late.
Okay, then next slide, please. You can even go in a developing floral meristem here and ask, are there differences between the adaxial, the abaxial, and the central domain of a flower before we have floral organ specification? And again, we can detect, during this kind of virtual microdissection, a number of genes which are expressed in this specific domain, which open them up for further analysis. Next slide, please. We can jump over this because there is a bit, we can also do that for the vascular system. And now, of course, we had in our lab, we were studying for many years stem cells and their functions in controlling many aspects of plant development. And I think you all know that, for example, in tomatoes, this increase in fruit sizes is to a large extent due to an amplification of carpel number, due to an increase in the size of the stem cell domain in floral meristems, which is by and large regulated by peptide and receptor pathways and by target transcription factors. I don't want to go into any details here at all. And these pathways are conserved. For example, in maize, if you knock out one of these receptors with restrict stem cell number, then the plants develop a larger inflorescence, a cob later on with a larger diameter and more kernel rolls. And this is work here from Dave Jackson's lab on the left-hand side. That is work from Zach Lipman's lab, and both are in Cold Spring Harbor. So that indicated that these pathways are highly conserved in plants. And go to the next slide, please. You will get this is just sort of an appetizer for a Barley Hub seminar, which will come by Isaiah Vadaniga in a few weeks' time. So he looked into a whole class of receptors which are involved in restricting or controlling stem cell behavior and also signaling between meristems, as we learned by now. The clavata-like receptors, so it's just a little bit of a screenshot on the left-hand side from alignment of these receptors between different plant species. We happen to have 14 of these genes probed on our single molecular RNA fish experiment. The overlap of that is shown here. And on the right-hand side, we picked out one of them, which looked most likely like clavata-1 from other species, and the expression pattern here. And we have expression information from most of the other receptors, which are related from our BARVISTA experiment. What Isaiah and several of the colleagues here in the lab did, next slide please, is then to follow our typical approach, and that is we were looking for mutants out there, but also generative mutants using CRISPR-Cas and gene editing. And here's one example from Isaiah, and he will explain that in much more detail. What he generated when he knocked out several of these receptors at the same time is to highly confuse the inflorescence development of barley. So we get a very confusing image here with lots of floral organs and tissues growing. What he did here is basically color code the tissues that he could identify. Well, how could he do that? He used the BARVISTA tool for that. So if you go on the next slide, please. We used the same trick as before. So he characterized generated, let's say, fingerprints for specific cell types or different differentiation status. And then fingerprinted in a similar way as a single mutant, double mutant, and triple mutant, and could thereby show that here there is an overproliferation in the triple mutant of this here light blue identity, which you find here at the base of lateral primordia in the wild type. And there is a lack of the magenta identity, which you find here in the wild type, in the developing floral meristems. And you find that here only occasionally. So we apparently have an increase here in meristematic identities, which we can basically map back to the wild type positions and therefore more clearly define our phenotypes. So next slide, please. Okay, that is basically just an eye pleaser. On the right-hand side is the output that you generate from the BARVISTA database. Next slide, please. And the last one is a bit of a group image from which is already some years old now. But I want to indicate who are the people that were doing all this work. And one of them is actually online today. I just saw. Hi, Edgar. So Edgar de Mesa Arevalo is now has an independent position in Mexico, and he was working jointly in my lab and in Maria von Korf's lab within our excellence cluster on plant sciences, and he developed most of these methods, and I will tell you who else helped him. Isaiah Vadaniga, he did, sorry, that is shifted somehow. He did a lot of work on these clavata receptors and signaling pathways, and he will report in a few weeks' time in a separate session. Jan Maika also contributed a lot to this and is still working together with Isaiah on the analysis of receptors and peptides that are communicating information within the inflorescence system. And none of this would have been possible without our incredible collaborators at the research center in Jülich, also within our excellence cluster. The informatics lab there is headed by Björn Usadel, and with a very, very talented PhD student who's close to finish now, Hannah Dörpholz, who set up the entire BARVISTA database and has now been joined by other colleagues, by Itzel, who's also contributing to this. And that was, all of this was a very close collaboration here with the lab of Maria von Kauf-Schmiesing, without whom none of this would have been possible. So thank you for your attention. And the last slide is indicating that meristem research in cereals is becoming has become very popular in Germany. So we decided with a group of people, which is funded by the DFG, in the cereal stem cell consortium, the group leaders are, their names are indicated on the bottom. We decided to have a nice international meeting focusing really on cereal meristems and stem cell systems at the University of Regensburg in Germany. If you are interested in that, that will take place in end of September this year, with a fantastic outline of invited speakers already. And if you want to come, please do. That's all. I'm ready for questions. Maybe we stop the presentation.
Thank you very much. That was incredible, as always. I'm not sure if people can unmute anyways and ask the questions, but I will just start from some questions that we have in the chat. Nupur Taylor is asking, he said, they say very interesting. And are the mutants information based on real data, or are they imputed?
No, they're based on real data. So in some cases we have sections and did a single molecular RNA fish, and in some cases we have mostly single cell RNA seq data. So, but they are based. You need some data to really anchor that properly.
Thank you. Nikolai Adamski says, great talk, thank you. How many mutants do you plan to add to BARVISTA? Also, good question actually. Could you comment on the cost and complexity of it?
Oh my God, the costs are killing us. So I think at the moment we are paying, I mean, including sequencing for a single cell RNA seq experiment is at least €2,500. And the single molecule RNA fish is, it depends on the company and what kind of deal you have. But it's in the thousands. So that's why, I mean, it was possible to do that here as a joint effort within the excellence cluster, because we basically, there was so much interest by so many groups that we thought, okay, we collect the money and afford that, to do that together. I think as a single lab, I wouldn't have been able to do that at all. And as I said, it's not my work. I'm just representing it here. It comes from a number of different people, and we pooled the money. But we hope to build it up as a resource for everybody. So we hope to have this online soon and get your feedback and your complaints, and then we can slowly improve that, or rapidly improve that, and add more data to that. That's the idea. And as I said, the other step is to connect that with cis element analysis, that we can really say, okay, this transcription or this gene is upregulated in my cell of interest, and I find at the same time, you know, transcription factor A, B, C, whatever target site has now opened up, so that we can really use that to build gene regulatory networks. And that is where we are still at the beginning.
Christina is asking, is there the possibility of adding a more complete 3D structure to the fish images?
Yes. I wish. we are actually, we plan to do that we do serial sections. And the next question that she should have asked, and will probably ask soon, is, can you do a time course as well? Yes, we hope to do that. But as I said, maybe for Christmas.
Okay, I think the next question aims at the same thing. So then Tanvir Khan is asking or saying that the data is highly relevant for meristem gene editing. Are you looking into that, and are you open for collaboration with industry?
Whether we are open for collaboration with industry, of course. It depends what kind of collaboration. Now, of course, these data should, we are happy to share our strategies there and what we do. I mean, the editing we do at the moment is as I mentioned initially, and Isaiah Badaniga will report on that in more detail, we started to edit the clavata receptors and related peptides. So we have a lot of edits already. We started initially with gene knockouts, and now we are also going for expression modification and so on, in order to see what kind of whether the barley architecture as we know it now is actually the end of it, or whether we can tune it in all possible other directions. So I was always promising here in the lab that we, in a few years' time, we will eat a barley cob and not a maize cob anymore. But whether that is possible is a different scenario. But I think at least editing a lot of genes and modifying them, tuning them up and down, will give us a much better understanding of how these inflorescences are, or complex architectures are so nicely coordinated and regulated.
Tanvir Khan says, thank you. Maybe one follow-up to a question from before. Someone asked about complexity. Like if anyone is thinking about establishing that for a new plant, a new tissue, how complex is the establishing such a method?
I mean, I would have thought it's incredibly complex, but I'm not a bioinformatician. I think you need a really good team, and we were lucky here to assemble a really good team of people who are good at cell biology, who have the guts to really establish methods. Thank you, Edgar. I mean, he did a lot of this work, and thank you also to the rest of the team here who are incredibly insistent on trying to make this single molecule RNA fish experiments work. So there was a lot of failures initially, and we got there where we are now. So I think you need a very good team. You need patience. You need shitloads of money, and you need a very, very interested. You need collaboration partners who are very, very good in bioinformatics and who are willing to donate years of their PhD life and resources to that. So if you have all this, then it's absolutely easy, you know.
Okay, I would rate it rather complex, but okay. I don't see more questions. Ryan, do you see anyone raising hands? Because I think people are only able to contribute to the chat.
Yeah, we've had one hand raise. So.
Ah, right. Okay, Rungshan, you want to ask a question?
Hello, it's a very impressive work. I'm sorry that I missed the first few minutes of your talk, but are you using a Visium system or?
No, that is the, in our opinion, the resolution of these systems in our hands. I'll be very, very careful because there's so many new developments on the markets. Okay. I step back. I would say we used molecular cartography at the time, which is a system that was pioneered by Resolve Biosciences, but there are many, many other systems around on the market which can be used for that. I think what sets our approach apart from a lot of the other colleagues around the world who are studying wheat, rice, maize, and so on, is that we really obtained cellular and subcellular resolution, a very high dynamic range, and I think that was beneficial for our experimental approach. I am not sure. We are testing out a few ideas at the moment whether we could actually simplify this drastically, but that is work in progress. We didn't yet try the Visium, but I've seen publications by colleagues that was also pretty good. There's actually, I've seen a new paper just coming out on bioRxiv on wheat, where they also combined single cell techniques with spatial techniques, and it's a wonderful work. And I think you can fill really pages and pages and pages with all kind of analysis that you do. But in the end, I think the best thing is if every researcher can just do their own analysis and look for their processes, genes, that they're most interested in. The rest is really super nice to see the potential of these methods. And I think we haven't seen the end of this. This is just the beginning.
Yeah, I agree. I also want to ask about some of the technical challenges. So we are looking at some, for example, a single cell nucleus data from 10x system,
Yeah.
right? So of course, a lot of barcodes are supposed to be empty. So, like, what is your definition of a good quality cell? Like, how many genes do you expect to be expressed in that cell? To say, you know, this is
I can tell you what we detected, and that was an average of, and Edgar would know it better if he can write it by heart. It was more than 5,000 cells which we found to be expressed. Sorry, more than 5,000 genes which we found to be expressed per cell. And I think the number of transcripts per cell, I would have to look it up. I don't know it by heart. Probably Edgar would know. I can't tell you at the moment, but I think that is the quality criterion, saying how many genes do you find expressed. And there are probably many, many more which you don't see because they're too slowly expressed. We know of a number of genes from reporter lines and from mutant studies. We know they are expressed in these cells, which we never detect in our RNA-seq experiment, whether it's single-cell RNA-seq, whether it's spatial transcriptomics or anything.
Okay. Wow, that's impressive. Thank you very much.
Most welcome.
Okay, we have one more question from the chat from Roberta Rossi, who asks, thank you very much for sharing your impressive work, and asks, when you indicated 48,000 genes, are you including alternative splicing variants in this?
No, we haven't really looked into that. That is really what we had from the number of genes basically comes from the latest annotation, which was done also by the USADA lab that used the Mercator tool, which was an in-house development some years ago for rapid gene annotation, and that's where the number came from. And we didn't look into alternative splicing. Wow, there are endless possibilities. Yeah. I also wish we could detect microRNAs and small RNAs and stuff, and that's all not yet possible, but I'm sure this will come in the near future. I'm absolutely sure that somebody has already some ideas on how to do that. Then we can do it all again from start.
Thank you. So we have some comments about how great the talk was from Sarah McKim and
Thank you, Sarah.
Yeah, and if we have no more questions, I cannot see any popping up in the chat at the moment. I think we would like to thank you again for this brilliant talk, for the brilliant work of the team.
The team.
For your time, yeah.
Not me.
You and representing your team.
Yeah. Thank you all for the opportunity. Good. And look out in bioRxiv. I hope the paper will be out very soon, and then you will have an access code to play around with BARVISTA yourself.
Sounds brilliant.
Good. Then
thank you so much.
Goodbye.
Bye-bye.